Well, good day to everyone joining us and welcome to today's X Talks webinar. Today's talk is entitled New Options for TQT Waivers from First-in-Human and Oncology Phase 1 Studies. My name is Maya and it's my pleasure to be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit questions and comments during the presentation and we'll try our best to attend them during the Q&A. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, you can contact me at any point by sending a message using this chat box. We also have 3 handouts for this presentation, including a PDF version of the slides. So attendees, you can download them through the handouts tab in your control panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is a leading healthcare research and technology company that generates the richest clinical evidence in the industry. Across decentralized, hybrid, and site-based clinical trials, Clario's deep scientific expertise, global scale, and the broadest endpoint technology platform allows the partners to transform lives. Now it's my pleasure to introduce our speakers for today's event, and our first speaker is Dr. Borya Darpo. Dr. Borya Darpo is the Chief Scientific Officer of Cardiac Safety at Clario with board certifications in cardiology and internal medicine. Dr. Darpo represented the European pharmaceutical industry on the ICH E14 Expert and Implementation Working Group between 2000 and 2008. In collaboration with industry and the FDA, he led the IQ-CSRC study, which validated the concept of applying exposure response analysis on data from early stage clinical trials to replace a thorough QT study. This contributed to the revision of the ICH E14 guideline, which since December 2015 allows this approach to replace the TQT study. In his current role, he supports business development and provides cardiac safety consultative services, advising sponsors on optimal cardiac safety strategy for their development program. And our next speaker is Dr. Robert Kleiman. Dr. Robert Kleiman is the Chief Medical Officer at Clario with board certifications in cardiology and cardiac electrophysiology. He has performed research in both basic cellular electrophysiology as well as clinical electrophysiology. Dr. Kleiman trained at the University of Pennsylvania and practiced clinical cardiology for 12 years before joining Clario in 2003. In his current role, he oversees all of Clario's cardiology services, supports business development, and provides cardiac safety consultative services, advising sponsors on optimal cardiac safety strategy for their development program. So now, without further ado, I'd like to pass control to our speakers. So Dr. Darpo, whenever you're ready, you can go ahead and get started.
Thank you so much for the nice introduction, and most welcome to all of you attending this, I think, interesting seminar in which we will discuss the new S7B E14 Q&A document issued last year in February that provides guidance on how we can replace the THERAQT study by implementing ECG monitoring, more specifically in first-in-human trials, and then support these data with non-clinical assays performed as per best practices, which also will be covered. I will cover the clinical parts of this, and Dr. Kleiman will then address the non-clinical assays. As you know, this is a highly regulated area. In 2005, the ICH E14 was issued and endorsed in all regions, and that document specified a designated study, the TQT study, in which sponsors were requested to perform to expose healthy subjects to high doses of their drug to ensure that the drug did not cause QTc effect of concern. Today, more than 400, more likely close to 500 TQT studies have been conducted. And I think it's fair to say that the average cost over the years has been around at $1.5 to $2 million US. In 2015, the E14 was revised. I should also mention that E14 has been the subject of several Q&A documents. And in 2015, there was a Q&A document issued that allowed us to use concentration QTc analysis to demonstrate that the drug did not cause a concerning QTc effect. This was much based on FDA's experience and sponsors' experience with concentration QTc analysis, but also on the so-called IQ-CSRC study. Now, most recently, in February last year, there was an S7B E14 Q&A document issued. So combining S7B with E14, describing how non-clinical assays, or more specifically the HERG assay and the non-rodent in vivo assay, can be conducted as per best practices, and how these non-clinical data can support a negative clinical assessment. So these are two examples of TQT studies. Lamidipine, both these are published. Lamidipine on the left side, where we can see moxifloxacin in the purple graph, and then the drug in green graphs, and ketoconazole in the middle. In the initial way to analyze these data, we were asked to exclude that the drug had an effect of concern at each and every time point. So at each and every time point, the upper confidence bound had to be below 10 milliseconds. That makes this a quite inefficient analysis, requiring somewhat between 36 and 44 subjects in a crossover or a randomized design. On the right side, we have a TQT study performed in schizophrenic patients, a bit unusual, with placebo response in the gray and the drug on day 13, combination of samidorphan and olanzapine in the green line and bars. And we can see here that the effect of the QTc interval above 10 milliseconds can be excluded at each and every time point post-dosing. And this then results in a clean US label. I give an example of this down below. So as I see it, the journey towards more efficient ways of looking at ECG effects or evaluating potential ECG effects started in 2015, when we were allowed to start using concentration QTc analysis to exclude the small effect. And as I mentioned, this was based on experience, but also based on a prospective study designed in collaboration between industry, regulators, and mostly FDA and academia. And today, in many first-in-human trials, ECG monitoring is paired with PK sampling, and those data can be used to claim that to replace the TQT study eventually in the clinical development. And of course, if you collect and store the waveforms from a first-in-human study, and then later decide that achieved concentrations were high enough to replace the TQT study, that becomes even a more efficient approach. It's also important to point out in those cases where we need to perform a TQT study, this can be done today much more efficiently than with a by time point analysis. With concentration QTc analysis, you need around 24 to 28 subjects to exclude a small effect of a drug. So this is one example from the IQ-CSRC study, and this is then dolasetron, or hydrodolasetron as the active mode is called. And we're looking at a concentration QTc model and the goodness of fit plot. The black line is the predicted effect of the drug, and the gray shaded area are the confidence intervals, or is the confidence intervals. The red bars shows that the model captures the observed data in a good way. And as we can see here, the slope of the concentration QTC relationship is positive. And we can also see that at the concentrations of a low dose on dancetrone in this study, around 200 nanograms per mil, we cannot exclude a 10 milliseconds effect. And therefore, this drug met the criteria that we had established prospectively to deem that we have detected a small effect. And this is then levocetracine, a negative study in the same negative drug in the same study. Bear in mind that we're looking at 9 subjects on active and 6 on placebo, so very, very small numbers. And with these numbers, it was possible to exclude that levocetracine had an effect of concern on the QTC interval. We can see somewhat wider confidence intervals, but even so, the upper bound is below 10 milliseconds throughout the observed range of levocetracine concentrations up to very, very high concentration levels. So for this negative drug, we were able to exclude that the drug had an effect of concern on the QTC interval. So in December 2015, E14 was revised, and there were 2 important points. The first one is that if sufficiently high multiples or clinically relevant exposure or concentrations are achieved in your first human study, then a separate positive control would not be necessary. The problem being that this was never defined. What is then sufficiently high concentrations? Then on the decision-making part of the guidance, it is stated that if you can exclude that a drug has an effect on the QTC interval exceeding 10 milliseconds at clinically relevant concentrations, then that is the same as to say that you have a negative QT assessment and you can replace the THERAQUT study by these data. And this is how it has looked then since 2015. The blue bars are the therapeutic studies, and you can see, and these are the proportional studies each year across an average of 55 studies per year. And you can see that the proportion of therapeutic studies have declined from 2015 and onwards. The red bars are what we today call the 6.1 pathway, which is mostly oncology studies, where you cannot give a high dose to healthy subjects. And we can see that the relative proportion of oncology studies or QT assessment in oncology studies has remained pretty much pretty stable. Whereas we can see that the green bars, which are the 5.1 pathway studies, which is drugs that can be given to healthy subjects in high concentrations, that proportion has increased and is still increasing, and will, as we see it, most likely replace a larger proportion of the therapeutic studies. The revised S7B E14 guidance from last year contains some very good definitions which we need to look at. So by a therapeutic dose is meant the highest recommended dose in the label. Clinical exposure is the resulting mean steady state Cmax after the highest recommended therapeutic dose. The high clinical exposure scenario is a term that will come back again and again. And by that, it is meant that Cmax at steady state, when the largest effect is seen by either a drug interaction or an internal factor such as renal impairment or hepatic impairment. So you need to define which patient group will have the highest concentrations, and the mean Cmax at steady state on the therapeutic dose in that population constitutes a high clinical exposure scenario. To give you an example in the right box, if we have a drug that is a 3A4 substrate, then we need to look at the geometric mean Cmax at steady state with this drug plus a potent inhibitor such as itraconazole. And that is then the high clinical exposure scenario. And this refers both to parent drug and abundant metabolites. This is one example that we published 2 years ago in collaboration with 4 different sponsors. So this is, as you can see by the abbreviation, a drug from Eisai. It's a phosphodiesterase inhibitor in development for dementia with Lewy bodies. Non-clinically, there was a high IC50 value of 4 micromolar. In non-rodent, in non-human primates, heart rate increase and also an increase in QTc values were seen at very high doses. And here we're looking at data from a SAD and the MAD study, so pooled analysis. And this, if you look at the left graph, you have the concentrations in the MAD part. And we can see that the drug substantially accumulates from day 1 on the left side to day 14 on the right side. So we have substantial accumulation, which means that the highest concentrations of this drug will be seen with multiple dosing. The right graph shows you the change from baseline QTc, with the black line being placebo, pooled from all dose groups. And the colored lines are the different dose groups. In the MAD part, we're looking at 4 different doses between 50 and 400 milligrams per milligram QD. And if we just look at the graphs here, you can see many time points at which the upper bound exceeds the 10 milliseconds. But even so, the pattern across dose groups does not suggest that this drug causes anything of concern. And this is how it then looks if we take the same data and analyze the data using concentration QTc analysis. On the left side, we have a scatter plot showing Japanese subjects in red and Caucasian subjects in blue. And you can see a very typical pattern with so much data on the low end, and in this case, relatively much data on the high end, but of course, not so much on the very high end. The right panel then shows you the goodness of fit plot of the concentration QTc analysis, with the black line being the predicted effect and the gray shaded area, again, the confidence interval. And we can see that only at the very high concentrations, up to around 3,600 nanograms per mil, we see that the upper bound crosses the 10 milliseconds threshold. Now, with this drug, the high clinical exposure scenario has been well defined and is around 1,300 nanograms per mil, which is almost 3 times lower than the concentrations at which we can exclude a 10 milliseconds effect. That means that there is no need in this case to perform or go back and look at the non-clinical assays. These data will be sufficient to waive the request for a surrogate study. So why do we place so much emphasis on the ECG methodology as such? And this graph, which I showed previously on levocetracine, illustrates this quite well. These data are generated from 9 subjects on drug and only 6 on placebo. And what matters here is the width of the confidence interval. The more precise ECG method you have, and also the more stringently controlled the experimental conditions are at the clinic, the more narrow confidence intervals you will have. So it matters which type of ECG methodology you work with. So we use in all our studies in healthy subjects, we use a technique called the expert precision QT, EPQT. So I will outline the main differences between the standard technique and the EPQT. The upper panel is a standard technique. We and most other core labs today would use a Holter recording, and from the Holter recording, from the continuous recording, ECGs are extracted. With a standard methodology, 3 ECGs are extracted, so 3 strips of ECGs. And in total, 9 beats from these 3 strips are analyzed. With a method that initially places the ECG complexes on screen, and then the technician either accepts or does not accept these values. So it's a mixture between a measurement made by an underlying algorithm and the technician/cardiologist. With the EPQT, we also use a Holter, and we extract up to 10 ECGs per time point. All beats from these 10 replicates are qualified or categorized into high confidence or not so high confidence. This is done by an underlying computerized algorithm. And the high confidence beats, the measurements on those are accepted, whereas the not so high confidence beats are looked at by the technician and either rejected or accepted. In a very, very small proportion of beats, there is some type of overread with adjustment. But we're basically looking at a median of around 100 to 120 QT pairs per time point. So much more data using one uniform methodology. And this then translates into precision. These are data from 7 therapeutic studies performed by Novartis some years ago and published. And for most of these slides, you will find the reference to the publication at the bottom of the slide. So we're looking at the variance with a by time point model, and the red bars being a standard technique, and the black lines and bars being the EPQT technique. And we can see that whether we look at test drug versus placebo, upper left, or MOXIe versus placebo, and whether we look at crossover design studies, upper panel, or parallel design studies, we can see that EPQT roughly halves the variance across all time points as compared to standard technique. And that means that we are more likely to exclude a small effect with EPQT than when we're using a standard technique. These are data based on simulation studies that we did internally, but also in collaboration with FDA. And if we assume a small underlying effect of 3 milliseconds, and if we then use EPQT, which typically would have a standard deviation or change from baseline QTc of around 6 milliseconds, then we need 9 subjects inactive and 6 to exclude that the drug has an effect of concern, that is an effect above 10 milliseconds. Using a standard technique that comes with a standard deviation of around 10, we need roughly twice as many subjects inactive with the same number on placebo. So it does matter which type of ECG technique you will use. So the background to the revised document is clearly that there is an increased interest in standardizing non-clinical assays. So far, the non-clinical assays, so the HERG and the non-rodent in vivo, has been used to make sure that the drug can be safely taken into man, so to ensure the safety of healthy subjects in the first in-human trial. At the same time, and over the last 10 years, FDA has promoted the CEPA project with the objective to improve the assessment of proteomic risk and to reduce clinical testing. And the CEPA hypothesis was that mildly QT prolonging drugs can be safe if the prolongation is limited to the latter part, so the JT peak interval is unaffected. So they've run a large number of research studies testing this hypothesis. Now, during the E14 or SFMB E14 ICH process, it has become clear that no other regulators have been really interested in taking on yet another not well-validated biomarker, such as the JT peak. But at the same time, both regulators and the industry has had the desire to further reduce the number of therapeutic studies. So overall, this has clearly increased the interest of non-clinical assays. So while the CEPA hypothesis and strategy has not been incorporated into the new guidance, it's fair to say that this whole discussion has promoted the interest of non-clinical assays, and even more so, the standardization of non-clinical assays. So the 5.1 pathway is when the drug can be given at sufficiently high concentrations to healthy subjects. So based on the 2015 revision, we know that if we reach a sufficiently high multiple, then we can replace the therapy study. This is now defined as being 2 times above the high clinical exposure scenario. So if you achieve concentrations, as in the example I showed you, that are at least 2 times higher than what will be seen in any patient group, then these data will be good enough to waive the surrogate studies. And you don't need to look at your hERG and non-rodent in vivo studies. But what is new is that when it's difficult to achieve this fold margin for the clinical assessment, so when you cannot reach twofold above the high clinical exposure scenario, then you can support your data by standardized hERG and a standardized standardized in vivo assay. And if these are both negative, then this combination of having negative clinical study covering what you will see in patients but not exceeding much with a double negative non-clinical assay assessment, then that can also replace the surrogate study. So this is another example shown in the same publication a year ago. So this is another compound in development, and it's clearly so that the PK characteristics in patients for this compound is not yet well shown. So these are still data from a first-in-human study. And on the left side, you can see this typical scatter plot with a quite nice distribution of data points from very low concentrations and up to high concentrations. And these data come from a SAD study. Then you have the model here, which shows you that you can exclude an effect about 10 milliseconds, actually above 5 milliseconds, up to concentrations of around 600 nanograms per mil. Now, it is not yet clear what constitutes the high clinical exposure scenario for this compound. So the sponsor then has two choices. Either go back and look at the hERG and in vivo study to see whether they fulfill the criteria set out in the S7b E14 Q&A document, or perform an additional clinical study adding higher dose groups. So those are the two alternatives. 6.1 covers drugs that cannot be given at high concentrations to healthy subjects. And this is not new. This is what we've been doing over the last at least 10 years, but it's better defined and better described today. So we are asked to take as many components from the therapeutic study that are feasible and implement these into a study looking at the CG effects. And this typically means that we're looking at oncology compounds and without placebo and without high doses, because the MTD dose is often used in oncology. The new item here defined in the S7b E14 guidance is that if you have a negative assessment in your clinical study, so at the therapeutic dose, you can demonstrate that the drug doesn't cause anything above 10 milliseconds. If you then support the clinical data with negative non-clinical data, then you can claim in discussions with regulators that the drug has a low likelihood of proarrhythmic effects due to delayed repolarization. This, of course, then also requires that you have a clean database. And also, there is probably some flexibility around this, as given in this sentence down below, that even if your clinical assessment isn't crystal clear below 10 milliseconds, there is still the chance for discussions. And this is also now better defined. This is not new, but it follows the practice that FDA has been using over the last years. If non-clinical studies are not done as per best practices, and you have a negative clinical evaluation, then the label will say something, at least in the United States, that the treatment is unlikely to have an actual mean effect as large as 20 milliseconds. Now, this is American labeling, and as an example, the Japanese label will not look this way. So this will be based on each region's decision, of course. And my last slide shows you a typical example of an oncology 6.1 pathway study. We're looking at data from 62 patients in part one, the dose exploration study, first in human study in patients with solid tumors. And we can see by using concentration QTc analysis for this drug, we can clearly see that the green shaded area is below 10 milliseconds. We can exclude that this drug has a large effect on the QTc interval. This was done at a time where the S7B14 guidance wasn't in place, so therefore the FDA label says that erdafitinib had no large effect on the QTc interval. And thereby, I would like to leave it to Dr. Kleiman, who will cover the hERG assay and the non-rodent in vivo non-clinical assay. Dr. Kleiman, please.
Thanks. Hi, folks. I'm Bob Kleiman. I'm a cardiac electrophysiologist and VP of Cardiology and Chief Medical Officer for Cardiology for Clario. And I'm going to talk about the non-clinical side of the S7B-E14 updates. And this is actually very important because this is the very first update to S7B since the release in 2005. And in fact, the part of the new document talking about S7B actually is longer than the original S7B release. Now, I'm not a safety pharmacologist, although I work with some safety pharmacologists, and for those of you interested, a very in-depth series of white papers about best practices for in vivo ECG studies is on its way. But I'm not going to talk about all of the details of that. Instead, I want to address what are the considerations someone on the clinical side of drug development would have based on these new pathways that have been made available to us. So at a high level, the new document provides a description of some best practices for non-clinical assays. And for our interest, those are first the hERG patch clamp studies, and secondly, the non-rodent in vivo cardiovascular safety studies. And both of these have to study both the parent as well as relevant metabolites. And the guidance documents go on to describe what is needed for the data from the in vivo study to be able to add something to the clinical data that's already been collected. And for the 5.1 pathway, the in vivo studies should cover at least the anticipated high clinical exposure. And for 6.1, it should also cover the high clinical exposure, but there's also language about the required sensitivity. The in vivo assay should have a sufficient sensitivity to detect a QTc prolongation effect of a magnitude similar to that in a dedicated clinical QT study. So some important information about how the studies need to be conducted. First, for the hERG study, best practices, and most importantly, that you should look for when you read the hERG channel study report are, one was the assay conducted at physiologic temperatures or room temperatures. That has a large impact on the results. And then were the concentrations of the parent or metabolites actually measured in the bath, because that is really what the cells will be exposed to. There's some discussion about use of concurrent positive and negative controls, and then discussion of the hERG safety margin. So the hERG safety margin is calculated as the hERG IC50 divided by the free concentration at steady state, Cmax, for the high clinical exposure. That's made very clear. This is not the free concentration for the clinical exposure, it's for the high clinical exposure. Protein binding obviously has to be taken into account to calculate the free concentration. And the safety margin that you calculate should be compared to the range of safety margins computed under the same experimental protocol with reference drugs with known QT prolongation effects and some risk of torsade. And the training materials have some nice examples of tables that can be used both to enter data that the regulators would be interested in when deciding if the non-clinical data are helpful here, as well as for our understanding of what they're looking for. Similarly, a summary of what would be expected for a 5.1 pathway. Now, for those of you who've never been involved with the non-clinical studies other than reading about them in an investigator's brochure, there are actually some very large differences in the conduct of in vivo safety pharmacology studies and clinical ECG studies. The first and largest of them is the number of subjects used. Most in vivo cardiovascular safety studies use a total of 4 animals. Sometimes that'll be 6 or a maximum of 8, but obviously that is far lower than the number of human subjects that we'll have for any clinical QT data submission. A thorough QT may have several dozen, and even an ascending dose study will have multiple cohorts. The measurements are also performed differently. Because the animals are freely moving, their heart rate is all over the place, there's a lot of motion artifact. So even though they use a single implanted telemetry lead and not a surface 12-lead ECG for the animals, you still have to average the ECGs over a period from minutes to hours. They don't extract standard 10-second ECGs as we would use in clinical practice. And the PK cannot be collected at the same time as the ECGs because the animals are freely moving. And when you restrain them and draw blood for a PK sample, their heart rate and autonomic tone go through the roof, and the ECGs are not useful. So you have to get around that in one of several ways. They may collect a single PK time point during the ECG portions just to correlate the specific results in that animal to what are expected, and then repeat a full set of PK assessments on a different day or even in a different trial. Unlike our thorough QT studies, the guidance has made it clear that you don't need to demonstrate assay sensitivity with a positive control in every in vivo ECG study, and there's discussion about how that would be done based on historical controls. Apologies, I am not a statistician and don't really want to spend much time on this. Just want people to understand that the guidance documents generally refer to the MDD, minimal detectable difference, as the statistical power for a study. But in reality, the MDD is a historical measure of the statistical power of a particular study design at a particular lab. It's based on prior studies. The LSD, or least significant difference, is really the study-specific sensitivity. So you have to be careful when reading the guidance documents about this, MDD versus LDD. And the guidance documents describe some of the best practices for in vivo cardiovascular safety studies. As mentioned, it's important to assess metabolites, not just the parent. And you have to consider the exposures that you're going to reach. And generally, you will want to try to reach at least the high clinical exposure. But you also have to be very careful about thinking about the limitations of the study, particularly given the small study size and the different conditions from a human study. You really have to look at the sensitivity and the statistical power to determine, are these data, even if, quote, negative, are they still going to be sufficient to add anything to what the regulators will see when they review the clinical ECG data? So when do these decisions get made about, can I use the patch clamp data and the in vivo ECG study data to support a 5.1 or 6.1 submission? Well, the answer is not at the time that the studies are conducted. The clinical data that are necessary to understand if the data are sufficient generally aren't known until well into clinical testing, often not until late phase 2, because you need to know the clinical dose and the steady state Cmax, both parent and metabolites, which means you have to characterize all the human metabolites. And you need to understand the high clinical exposure, which means you have to understand what happens in the presence of meal, drug-drug interactions, hepatic or renal insufficiency. So it's really only once the data from the clinical trials tell us the high clinical exposure and the clinical exposure that we can really begin to look and see, did the in vivo study data provide any data that are above the data obtained in the clinical trials. You'll also need to understand protein binding may be different between the animal species that was used and humans, and of course, parent and metabolites. So now you're at the end of phase 2, and you're looking back at your non-clinical data because you weren't in your first in human studies, you reached a high clinical exposure, but not twice the high clinical exposure. So you're going to look at your non-clinical data, and particularly the in vivo ECG study. And first, you're going to look at the bottom or top line. Was there a QTC increase detected or not? If there is, that's not necessarily a fatal flaw. If there's a QTC effect, first, where there are any extenuating circumstances and alternative explanations besides a real effect on cardiac repolarization. And in general, if you have QTC increases, but only at extremely high exposures, that may not be an issue. If you have a significant QTC effect at or below the high clinical exposure, though, that means a 5.1 option isn't viable. And if you see the same effect at or below the clinical Cmax, then obviously the 6.1 option won't be available either. But if you see only a small QTC increase at high exposures or no QTC increase, the next thing you have to do is really examine the study conditions and understand the sensitivity of the study and not just the mean effect, but the upper and lower confidence bounds around that. You have to take into account the sensitivity. If you test only to the high clinical exposure, then for that data to tell you something beyond what we've learned from the clinical ECG data collected, you need to have an excellent study sensitivity, an LSD of 3 to 5 milliseconds. Whereas if you have only standard sensitivity, an LSD of around 10 milliseconds, that means that a quote negative study that did not demonstrate a statistically significant QTC effect may not have excluded a clinically relevant QT effect. And that's really what counts. So in that situation, you would probably need a higher exposure than just the high clinical exposure itself. You will need to exceed that. And if you have just average sensitivity, an LSD of 10 to 20 milliseconds, you may need very high exposures to be able to exclude an upper confidence bound of 10 milliseconds for a QTC increase. Fortunately, high exposures cover a multiple of sins. The Q&A describes a bit about the sensitivity required for a 6.1 assessment, but it does not specifically state what sensitivity is desirable for a 5.1 assessment. But again, I think that based on common sense, we can guess at what will be going through regulators' minds when they look at the in vivo study data for a 5.1 submission. And remember, if all the non-clinical data does is show exactly the same data as the clinical ECG studies and doesn't supplement it, it's really not going to add in an integrated assessment. So as I said, it's important to look at the sensitivity of the study. If your sensitivity is very good, LSD or MDD of around 5 milliseconds, it may be sufficient to reach the high clinical exposure, but the lower your sensitivity, the higher the multiple of that you will need to achieve with a negative study to complement what we already know from the clinical data. As I said, the guidance does specify the sensitivity for a 6.1 scenario submission should match what's used in clinical QT studies. And the reference here does cover considerations of sensitivity. It says if the minimal detectable difference might be 5 milliseconds, the drug exposure probably only needs to cover the high clinical exposure, but might be higher if a larger multiple of the high clinical exposure is achieved. So if you show no QTc effect at 3 times the high clinical exposure, a sensitivity of 10 milliseconds might be acceptable, whereas that wouldn't be if you had only tested to the high clinical exposure. And unfortunately, I have to bring up an unresolved issue that was brought up in the guidance training materials, the relative cross-species sensitivity differences to hERG blockers. And the ICH training material states that in a dog, at any given exposure, moxifloxacin produces only 1/3 the magnitude of QTc increases in humans. So in dogs, you see a 10 millisecond increase at a plasma concentration of 12.6 micromolars. You see the same 10 millisecond increase in humans at a plasma concentration of 3.6 micromoles. The ICH training material sort of implies that that means you might need 3-fold multiples of human exposures for any particular value of QTc prolongation. And published data show that the same thing is true for other drugs and other species. So I think that this is going to be an issue. How regulators will view this is not entirely clear. There's some implications that because of this relative difference in sensitivity to hERG channel blockers, higher multiples of the high clinical exposure might be needed. And again, the training materials have more information about what are the data you need to collect, how, once you've looked at the data, can you decide, does this really supplement the data from the clinical ECG studies? So what do we think the impact of this new Q&A document will be? Well, clinically, I think it's going to be very important. As Dr. Darpar already indicated, we think that the proportion of new drugs that will be required to undergo a dedicated thorough QT study is going to continue to decrease. It's a little more difficult to predict the effects of 6.1, which really, in effect, say that if you have excellent non-clinical data, that may allow you slightly different labeling language. Not clear if all regulators will view that the same way, and not clear how advantageous that is. But at least for the 5.1 options now available, that is likely to make a very large difference in how we conduct QT assessments. On the non-clinical side, I think that the labs and sponsors are really going to have to think about when to use best practice studies, because that may be a deviation and more expensive than standard non-clinical trials. When will that be appropriate? Is it for all drugs? Is it only for drugs where one thinks that 5.1 or 6.1 might be appropriate? And I think we will learn that over the next few years. So to conclude, early QT assessment, as Dr. Darpo discussed, is now a fully accepted way to confidently exclude a clinically relevant QT effect for a new drug. And the revised Q&A documents are going to further encourage the use of early clinical studies to avoid the need for dedicated thorough QT trials. In order for us to use the non-clinical data, first, those of us on the clinical side of drug development are going to have to better understand the non-clinical data and learn how to determine were the studies done according to best practices. And labs are going to have to make sure that they are capable of following the new best practices being outlined. We don't think the TQT study will completely disappear. There will be some circumstances where it's still the only viable option. But overall, we think that this shift is going to make ECG and QT assessments more effective, more efficient. I think we're going to learn whether or not new candidates have potential QT risks earlier in development. And we would hope that ECG assessments can be performed as a routine part of SAD and MAD studies for new compounds and avoid many times the need for a thorough QT study. And I'm going to stop there and thank everyone for their attention. We left a few minutes for question and answers.
Thank you, Bob. Thank you so much. We have more than 20 questions, so I'll try to select as many as we can over the next 6-7 minutes and see what we can address. Please bear in mind that we will provide you with answers on all questions after this webinar, and you can also reach out to both Dr. Kleiman and myself with additional questions. So, Bob, a question to you. What is the primary reason for FDA denying a thorough study waiver in your experience?
That's a very good question. The first reason out of two is that the request was made too early, and the FDA simply did not have all of the clinical data that they needed to make that decision. For instance, the high clinical exposure wasn't yet known. That's the case. You can't judge whether non-clinical data and the clinical data are sufficient. The second reason I would say is related to not adequately assessing either metabolites or accumulation for drugs with long half-lives. You have to assess steady state exposures, both apparent and any relevant metabolites. So those are the two most common situations I've encountered. You may have seen some others before you.
Next question is, how many subjects would be needed to declare a new drug is safe? That is to exclude a 10 milliseconds QTc effect. And that question isn't really possible to answer because it depends on the model, on the concentration QTc model. But we normally do not advise on the design of first-in-human studies because there are so many other very good reasons to perform a first-in-human study. But we recommend implementing the procedures. And if you end up having, let's say, one dose group at high concentrations and one dose group with really high concentrations, given that most of these studies are done with 6 on active and 2 placebo, then you are in many cases, if the drug is negative, you're fine. The data will support that you can exclude a small effect. Another important item is the number of placebo subjects. If you only have, we pool the placebo subjects in the analysis, but even so, if you end up having 4 or 5 or 6 placebo subjects, anything that will be seen in the placebo group drives the effect on active. So we really strongly advocate making sure that you include all those groups to also have the right number of placebo subjects. So the next question is whether there is a requirement to look at more than one factor that may increase Cmax at steady state. As an example, if both food and the 3A4 inhibitor increases Cmax. And actually, there is no such combined criteria. You need to look at you need to look at the scenario where you reach the highest mean Cmax at steady state, be it on 3A4 inhibition or renal impairment, but you don't need to combine these different scenarios, and that's something that makes life a bit easier. So Bob, perhaps you can also take a question in terms of the clinical assessment. What if cardiodynamic data are collected and in the SAD portion you have a 10-fold safety margin or really high safety margin as compared to the high clinical exposure scenario? Wouldn't this be enough?
We encounter that scenario, and the answer is sometimes it's enough and sometimes it isn't. So if you have a drug with reasonably normal PK characteristics and you've achieved 10-fold the high clinical exposure in your SAD, it's very unlikely that even with modest accumulation that you would be below twice the high clinical, a twofold multiple of the high clinical exposure. But if you have a drug that has very long half-life and very significant accumulation, or metabolites that have long half-lives and accumulate, the data from day 1 of dosing may not be sufficient to really characterize adequately high steady state exposures. The half-life's very short and no metabolites, you usually end up using just the SAD data. But if you don't know, what I always recommend to clients is you're going to collect in your MAD study PK data, not just at cycle 1, day 1, but at some later date at steady state. Since you're already collecting the PK data, I recommend that you have subjects wear a halter on that day, the second day of intense PK collection, and hold on to that data and see if the PK shows you that you need to have that ECG data analyzed and used as well.
There are several questions in terms of QTCF versus QTCI. And one question is that in one of the examples you gave, the test drug increased the heart rate. The analysis was done using QTCF. Why didn't you use QTCI? And the simple answer on this question, which is much more complex, but the simple answer is that if you don't have what is called a substantial heart rate effect, then you can stick with QTCF because it's good enough. It may not be perfect, but it's good enough. And with a substantial heart rate effect is meant a placebo-corrected change from baseline above 10 beats per minute. So if you have a more than 10 beats per minute effect, then you need to look at QTCI. Now, in most first-in-human studies, there is no day -1 ECG collection, and we don't typically recommend the sponsors to implement this either, unless they have a pharmacological action of the drug or has seen in non-clinical in vivo studies that the drug clearly has a heart rate effect. Then we recommend having a day -1 to enable exactly what you're asking for, QTCI. But we don't just say that QTCI is always better. We actually test whether QTCI removes the heart rate dependence better than QTCF or a population-based QT correction method. So it's a bit complex, and it's not always easily implemented into first-in-human trials. Bob, how strong is the evidence that a 10 milliseconds threshold distinguishes between safe and not safe drugs? Can this be redefined looking ahead, looking forward?
Thanks for that very easy question, Borje. Actually, Borje can probably tell us more because he was part of the original 2005 discussions about what should go into the guidance. And what's written in the guidance is the threshold of regulatory concern is a QTC increase of about 5 milliseconds as evidenced by an upper confidence bound of 10 milliseconds. And that would trigger enough regulatory concern that during phase 3, regulators would expect that you would continue to do more intense ECG monitoring because obviously patients might have more factors leading to QT prolongation risk than healthy subjects on other meds. So that's the original intent. 10 milliseconds is not, especially an upper confidence bound, is not meant to be, this is the cutoff above which all drugs are torsades. It was meant to be a yardstick for perhaps increasing scrutiny in late phase. Realistically, there are many drugs on the market that have QTC effects with an upper confidence bound between 10 and 20 that are used extremely widely and extremely safely. So 10 millisecond upper confidence bound, even in your clinical studies, doesn't mean that the drug isn't safe. It simply may mean that a little bit more caution is needed in use.
So I will address 2 quick final questions as people are dropping off. And the first one is to which extent other phase 1 studies, such as drug interaction studies, can be used and pooled to first-in-human trial data. to demonstrate that a drug doesn't have a concerning effect. And in principle, you can pool studies if you use the same ECG methodology, and importantly, if you have placebo in both studies. So DDI study is typically not conducted with placebo. You have drug and then the inhibitor and then drug plus inhibitor. So you don't normally have placebo. That makes it more complex. There are also other reasons for making DDI studies not so suitable for concentration QTc analysis to demonstrate that the drug does not have an effect, such as an altered parent metabolite ratio. And finally, the 10 extracts we look at when we use EPQT are those automatically selected. Well, we have an underlying algorithm that categorizes beats into high confidence and not high confidence. That is certainly a computerized algorithm that makes that first distinction. And by that, I think we should summarize or conclude that this is what we have time to cover today. And I would like to thank you so much for your attention. And also, again, if you have questions, please reach out to me or Dr. Kleiman. Be more than happy to address those. And also, all of you who have asked questions, you will receive answers eventually. Thank you so much from my side.
And thank you as well. The copy of the slides will be available as well, I believe. And there'll be some other information in them that may answer some of the other questions as well. So thanks everyone for your time. Thank you, Dr. Darpo, for a wonderful presentation as well.
Well, thank you very much. And just like Dr. Darpo and Dr. Kleiman said, if you do have further questions, you can use the email addresses on your screen. So go ahead and quickly jot these down if you haven't already. And thank you again, everyone, for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. Should take about 1 to 3 business days, so be on the lookout for that. A survey window will be popping up on your screen. Your participation is appreciated as it will help us improve our webinars. Now, please join us in thanking our speakers today, Dr. Borja Darpo and Dr. Robert Kleiman, for that fantastic presentation. We hope you found this webinar informative. Have a great day, everyone.